Good morning, everyone. It's a pleasure to be here this morning on this episode of Crops TV. My name is Alison Robertson, and I'm a professor and extension field crops pathologist here at Iowa State University. And this morning, I'm going to be talking about some corn disease research that my lab has been involved in the last couple of years. So some of the research that I'm going to be talking about are our corn fungicide trials. So these are the trials that I do every year where I compare timing and products around the state. And I will also share some data from some fungicide trials that we've been doing to manage Physoderma. And then most of my presentation is going to be on our cover crop research that we've been doing. And so I'm going to be talking about our planting green experiment and then also our social distancing experiment. Up there in the top, you can see a picture of my lab, and then Jyotsna and Sarah in the lab assessing corn disease seedlings, and Sarah taking soil samples at one of our farmer fields. And you're going to be hearing Jyotsna and Sarah's name come up a lot. So first of all, let's talk about my corn fungicide trials. So these are the trials that I affectionately call the cardinal trials. And you can see that I do them at 6 locations around the state. I think they're familiar to a lot of you out there. And the questions that I'm hoping to answer with these questions is, which fungicide controls disease best? I only look at commercially available fungicides, so those fungicides that are on the market at the moment. I'm also interested in, is there a fungicide that gives the best yield bump? And then my third and final question is, what is the best time to spray? And that's in terms of disease control and also for yield. So just a reminder about the 2020 growing season. So these are 2 maps. One shows the departure from normal temperature, and this is between the 1st of June and the 31st of August. And the other map shows the percent of normal precipitation for the same dates. And the reason I chose those dates is because that's when we tend to start seeing diseases start to appear on corn and then also just increase. So you can see from Iowa, probably don't need to remind many of you who live in Iowa, but for those of you outside of the state, we had a warmer than usual growing season, particularly in northwest Iowa. It was even hotter than usual. And then we also had a drier than normal growing season. So a drought across much of Iowa, particularly in western Iowa. In eastern Iowa, started off kind of wet, but then dried up as the growing season progressed. So, of course, you can imagine that with warmer than usual conditions and very low precipitation, we had virtually no disease. As a plant pathologist, it was a pretty boring season. So when I went out to these trials and assessed disease on the ear leaf, at all those locations, there was less than 1% of the ear leaf had disease. Most of that disease was gray leaf spot. There was a little bit of tar spot in the eastern locations. And then also we saw some southern rust, but overall less than 1% on that ear leaf. So I tested 2 fungicide application timings. One of them was the V12 application, and this shows you the yield response at each of those various applications. And you can see the fungicides that I tested along the x-axis and then the yield response. So, you know, that yield response ranged all the way from about -9 bushels to almost 8 bushels. So a typical range that we see in these yield responses. I also highlighted some data from the Northwest Research Farm and then also from the Agronomy and Ag Engineering and Agronomy Farm here in central Iowa in orange. And the reason why I did that was because I wanted to show you that, you know, at some locations, a fungicide didn't do very well. There wasn't a yield response, and yet in another location we had a very good yield response. And so there's not a consistent yield response across all these fungicides at V12. And the mean yield response we saw was negative 0.7 bushels per acre. So really, there was no yield response from that V12 application. If we look at the yield response for the R1 applications, this graph is laid out the same way. In this case, I've chosen the Northwest Research Farm and the Northeast Research Farm. But once again, you can see that a fungicide that did very well at one location did not necessarily do well at another location. And the mean yield response here was just over 1 bushel per acre. So really, in these drought conditions where we didn't have a lot of disease, we didn't see much of a yield response over all those data points. So now I'm going to move on to my fungicide trials that I've been doing to have a look at Physoderma brown spot and node rot. And in this trial, what we had was 2 hybrids. We had one hybrid that is susceptible to brown spot on the leaves, and you can see that hybrid on the left. And if you look down the center row of that plot, you can see the yellowing on the leaves, just showing the brown spot. This hybrid does not get node rot. And then we have hybrid 2 on the right, which has very little brown spot on those leaves, but is very susceptible to node rot. I call this a canary for the node rot. We did these trials at the Northern Research Farm at Kanawha. They have a field there that has a history of severe Physoderma, and so we were able to rely on natural inoculation there. And what we did is we had various fungicide treatments. So we had a couple of treatments from FMC, which is the Xyway product in the furrow, and then we had a half rate of Xyway in furrow with top guard at V6 to V7. The reason why the top guard's in the light gray is because we forgot to spray that. So really, we have a full rate and a half rate of Xyway in furrow, and then we have Dolaro from Bayer applied at V3, V4, V6, V7, V9, V10, and V16 to V18. This chart shows you the percent incidence or severity. So the leaf sheath incidence is shown in blue, and this is the number of plants on which we were able to see Physoderma brown spot on the leaf sheaths. And then the brown spot severity is shown in the orange. And so this is that brown spot severity, how much of the canopy was affected by brown spot. And so we have the check, and then we have the fungicides along the x-axis. And what you can see is that fungicide application at V6, V7, and then also at V9, V10 significantly reduced the amount of leaf sheath incidence that we saw, and then also the brown spot severity. And interestingly, the Xyway in the furrow, although it didn't reduce the number of plants on which we saw leaf sheath, we did reduce the amount of brown spot that we saw in the canopy. So that was very interesting. This trial we will repeat again next year, and we will try and tease out those timings between V5 through to V10 and try and have a better look there and see how those timings can help. So, but it does look like an early application of fungicide can help reduce the amount of brown spot in the canopy. One thing to note is that we did not have any node rot in this trial, and therefore, unfortunately, we were unable to tell if a fungicide application would have reduced the amount of node rot. So now I'm going to move on to talk about my cover crop trials that we've been doing the last couple of years. And the goal behind these cover crop trials is to optimize yields of corn planted after a cereal rye cover crop. And this research has been funded by the USDA NIFA, the Iowa Nutrient Research Center, and then Sarah also got a student grant from the North Central Sustainable Agricultural Research and Education program. And in these trials, we've also, apart from our small plot trials, we've also been working with the Iowa Soybean Association On-Farm Network and Practical Farmers of Iowa so that we're able to get onto commercial farmer fields and do similar experiments. So the reason why we got into these trials was because of this reported yield drag following a winter rye cover crop. And we wanted to understand what was causing that yield drag. And there's been a number of different reasons proposed for this yield drag. So one of them is poor seed to soil contact. And so that results in reduced stands and more barren plants. Allelopathy has had a finger pointed at it. We know that allelopathy can inhibit germination. And then also slow temperatures because of that rye residue, keeping those soils cooler, that can slow emergence. But the thing is, when I look at those reasons, what I think about is seedling disease. So if we look at corn seedling disease reduces stand. Corn seedling disease slows emergence and reduces vigor. And corn seedling disease can lead to barren plants because of these plants that are behind in emergence and then become weeds later on during the growing season, and so either produce a smaller ear or no ear at all. They're barren. So the other thing to remember about seedling disease is that that seedling disease root system is very, very important for seedling health. Okay, so when I'm talking about that seedling disease root system, I'm talking about the radicle that appears first, and then we have the seminal roots and the mesocotyl, which is the pipeline that carries those nutrients for that young developing seedling to the crown so that that seedling can grow. And then you can see there the development of those nodal roots. Those nodal roots are not fully active until we get to V6, but from about V3 to V4, they start to emerge and start to take over the nutrition of that seedling. So up until V3, V4, the seedling disease system is very important for seedling health. So let's get back to my question. Could seedling disease be responsible for yield drag? And I wanted to take you back to some early work that we did looking at this. So first of all, we asked the question, is winter rye a green bridge for seedling disease? What do I mean by that? Well, first of all, we found out that winter rye was a host of pathogens that cause seedling disease on corn. So Pythium and Fusarium also that can survive on winter rye and cause seedling disease on corn. So if what we hypothesized was that when you terminate rye in the spring with glyphosate, for example, you can see the Pythium populations, as those roots die, they release all this Pythium and Fusarium into the soil, and so we get this bump in the population. And then if there's no host, that bump in the population then declines and we go back down to our background levels of Pythium. So if we come in and plant corn when we have that high level of Pythium, we're more likely to get seedling disease. And that seedling disease would be radical rot or mesocotyl rot or rot on the seminal roots. So this would be the green bridge effect, right? If we wait and spray, if we wait until that population has decreased and then plant our corn, then we're planting into a lower background level of Pythium and we're more likely to get healthy, vigorous corn seedlings. So we did this work back in 2015 and 2016, and this was work done with funding from the Iowa Nutrient Research Center. And so what we did was we had our no rye, which was our control, and then we sprayed the rye at different days before planting. So 25 days before planting, 17 days before planting, 8 days before planting, 3 days before planting, and in 2015, 2 days after planting. And if we look at the amount of radical rot that we had, you can see that where we spray the rye very close to planting the corn, we had significantly more disease than we did when we sprayed the rye 17 days or more before we planted. Of course, it's important to see what this does to yield. So these are the same treatments showing you with the yield. And so you can see in 2014 that the rye that was sprayed 1 day after planting in 2014 had the lowest yield. But other than that, there wasn't significant differences in the yield, although the no rye check still had the highest yield. And then in 2015, you can see that there was a decrease in yield from our no rye check all the way down to that rye that was terminated very close to planting that corn. We got less and less of a yield. And one difference in 2015 was we had very high biomass. So based on this research, we came up with the recommendation that you should wait at least 10 days to plant corn after terminating your winter rye cover crop. But as many of you know, a lot of times in Iowa, we can have cold, wet springs. And those cold, wet springs prevent access to the field to terminate the rye. And also, when it's cold and wet, glyphosate is not as effective. And so, a lot of farmers, what that comes down to is either you can wait and wait until it dries up, spray the glyphosate, and then plant your corn later, but then you run the risk of later plantings and lower yield potential. Or, as some farmers are doing, you can plant your corn and then spray the rye a few days later. And this practice is referred to as planting green. But based on our research that we'd done, we had lots of concerns and questions about this practice. And so to figure this out, we got funding from USDA NIFA to answer this question: could planting green increase seedling disease and contribute to yield drag? So for this project, we had several research activities. We had an experimental plot field trial that was done in 2019 and 2020, and I'm going to show you results from that today. We also had some microplot studies. I won't be talking about those today. And then we have some on-farm research going on in collaboration with Practical Farmers of Iowa. So our first year for on-farm research was 2020, and we'll be following this up in 2021 with another year of on-farm research. So let's look at this experimental plot field trial that we had. So these are our treatments. We had where we terminated the rye 18 days before planting the corn. And you can see we had 2 treatments there. The nitrogen varied. I'm going to come back to that. So we had 18 days before planting, 3 days before planting, 6 days after planting, 12 days after planting. And then, of course, we have our no cover crop control. Then, as regards to the nitrogen, treatments 2 to 6 all received 50 pounds of nitrogen at planting and were side dressed with 130 pounds of nitrogen at about V4. Then we had, for our first treatment, what we did was that's where we terminated the rye 18 days before planting the corn, and at the day of termination, we also applied 120 pounds of nitrogen. And then we came back at V4 with a side dress of 60 pounds of nitrogen. So all treatments received 180 pounds of nitrogen. We just had slightly different timing of when that nitrogen was applied. And then the trials were set up on a corn-soybean rotation. This is a no-till field, and the rye was drilled in late September 2019 and 2018. We managed to get the rye in very good timing. So if we think about my hypothesis with the different Pythium populations, so in the brown there, you can see where we've terminated the rye 18 days before planting, the Pythium population increases, and then it decreases. Then you can see the Pythium population, how it might change 3 days before planting, 6 days after planting, and 12 days after planting when that rye is sprayed. And then I've put a red arrow there so that where you can see when the corn is planted. And so what we're hypothesizing is with 18 days before planting, we're not going to see a lot of seedling disease. But where we terminate the rye close to planting, or 6 and 12 days after planting, we're going to be planting that corn into a petri dish of Pythium, and therefore we're going to see more disease. So that's what we were hypothesizing. That's what we needed to collect our data on. So this is our this shows the plot field trial data. I'm not going to show you all the data, but just some of interest. So this is the rye biomass in 2019 and 2020, 2019 in blue and 2020 in red. And the first thing that you'll notice is that the later you terminate the rye, the more biomass you get. Kind of a duh statement, but the longer you leave that rye to grow, the more biomass you're going to have. But the other thing that I want you to notice as well is that in 2020, we had significantly more rye biomass compared to 2019. And that's just because we had a nice warm spring in 2020, which really helped with the growth of that rye. Okay, so let's talk about some of how those treatments affected early corn growth. So here you can see the 2 years, 2019 and 2020. You can see my treatments. No rye, that 18 days before planting with the pre-application of nitrogen, and then 18 days before planting, 3 days before, 6 days after, 12 days after with that nitrogen at planting. So if we look at plant height, in both years we detected a treatment effect. So remember, if a number has the same letter next to it, it means that it is not different from a number with the same letter next to it. But what you'll notice in this is that in general, the corn plants were shorter, where we had no rye or the rye was terminated early, compared to where the rye was terminated late. Those plants were much taller. And that's probably because they were etiolated, right? You can see how much residue was there in that photo that I have there, with that 12 days after termination, 12 days after planting, terminating 12 days after planting treatment. And so the plants were etiolated as they grew through all that residue. In 2019, we didn't see any effective treatment on growth stage, but in 2020, we did notice that even though those plants were taller, in where we terminated the rye later, they were actually a growth stage, a little bit behind in terms of growth stage. And when we look at the dry weight, we can see that those plants were also smaller. And so in general, what this is telling us is that when we plant green, the corn in this trial tended to be less vigorous than when we terminated the rye 18 days before planting, or compared to our no rye control. So of course, seedling disease is what I'm most interested in. And so this gives you an idea of what went on with seedling disease. So if we look at the radical severity, you can see in both years there was a treatment effect. And in general, what we saw was more disease when we terminated that rye around about or after we'd planted the corn. There was no difference with the seminal severity in either year. This chart here shows you the number of barren ears that we counted for each treatment. And so we have along the x-axis, we have the various treatments, no rye, and then the time that the rye was terminated. And what you'll see is 2019 data is in the light orange, and there was no significant effect or no difference in the number of barren areas between those treatments. And then in 2020, in the dark orange, we did detect some effect of treatment. And so what you'll notice is, interestingly, in the 18 days before planting, where we had the pre-plant application of nitrogen, that's where we saw the most barren ears, and then the least barren ears in that 12 days after planting. If we look at the yields now, So 2019 is in the light blue and 2020 is in the dark blue. In both years, we saw an effect of treatment on the yield. And so just as we expected, if we think back to this follows the disease data very nicely, is that our no rye check had the highest yield in both years, and then where we terminated that rye after planting, where we planted green, that's where we got the lowest yield in both years. And perhaps most surprising was that despite having very low vigor seedlings at V3, V4, and then having the most barren plants, that 18 days before planting with pre-plant applied nitrogen gave us the highest yield. So we're still trying to figure that out. And if any of you has an idea why that might have happened, we would love to hear those ideas. So apart from these small plot trials, as I mentioned, we have been doing on-farm trials with practical farmers of Iowa, and we've done these at 4 locations in Iowa. We have 6 to 8 strips in each of these commercial fields with a previous crop of soybean. And we're comparing 2 treatments where the rye is terminated 18 days before planting, and then the rye is terminated 6 days after planting. And here we can see one of our collaborators, Kevin Hall, with Giotsna, sampling seedlings from those plots. I'm not going to show any more data from the on-farm trials with you. The other thing that I want to mention is a project called Precision Sustainable Agriculture, and this is another USDA NIFA project that I'm involved on. And this is starting in 2021 will be our first year of collecting data, and it's almost a repeat of the planting green treatment that we did here in Iowa. But the exciting thing about this project is that it's going to be done at numerous states across the U.S. And we're not only going to look at the effect of cover crop termination timing on disease, but we're also going to collect data on slugs, on insects, pests and beneficials, and then weeds, and then obviously the corn performance. And so this map gives you an idea of where those trials are going to be. We have identical trials throughout the U.S. I am leading the seedling disease part of those trials. John Tooker from Pennsylvania is leading the insect collection data, and then Colleen Chase from Florida is leading the weed part of those trials. And so over the next 3 years, we will be looking at the effect of planting green on pest management. Okay, so the trial that I showed you, the planting green trial, is basically about using timing to reduce yield drag. But we had another question, and this was based on some work that Tom Casper and Keith Kohler did a decade or more ago. And our question was, could social distancing reduce seedling disease and minimize yield drag? And we started this experiment back in 2018, before any of us had even heard of social distancing. And so it's kind of ironic that we were doing this trial. But basically what we did was we started by doing a trial in the greenhouse. And what we did was we planted rye on one side of the pots. And then on the other side of the pot, we had no rye. And then we terminated that rye and planted corn seed into the rye residue, and then corn seed 4 inches away from that corn residue. And this just shows you a picture. You can see that the corn that was planted into the corn residue had rotted radicals, whereas the corn that was planted 4 inches away from that rye residue had healthy roots and basically escaped disease. So this was just done in the greenhouse, and so we were interested what would happen if we took it out to the field. So this is out in the field, and this is research that was funded by the Iowa Nutrient Research Center. And also Sarah got this grant from North Central SARE to fund her on-farm research. But basically what we have in this activity is we have an experimental plot field trial again. and we have a no cover crop check. And then we have a treatment where we broadcast the rye and then planted the corn into it in 30-inch rows. And then we have rye, which is planted 7.5 inches from the corn row. And so what you can see from the slide there is that we have 3 rows of rye, 7.5 inches, drilled 7.5 inches, and then we blocked off the 4th row, and that's where the corn's going to plant. So we have 3 rows of rye, and then we have where the corn will be planted, and then we have another 3 rows of rye. And so it ends up that that corn row is about 7.5 inches away from the rye. And then we have a 4th treatment where we drilled the rye in 30-inch rows and then planted the corn down the middle of those 30-inch rows. So the rye is approximately 15 inches from the corn row. The photo that I'm showing you here is from our 2020 trials, and so you can see we were a little bit off on our corn row, but in 2019 we were perfectly between those 2 rye rows. And then we're also working with the Iowa Soybean Association on-farm network, and we have a couple of field locations there that we've been working with them. So you can see Jyotsna and Sarah on the left, and a couple of our undergraduates out there at a farm collecting data. So I'm going to talk a little bit about the experimental plot trial that we had. So first of all, we'll start with the rye biomass. And we have 2019 and 2020 data. And then we have our no rye control, so no biomass, obviously. And then we have the broadcast, or the 0-inch, 7.5 inches away from the corn row, 15 inches away from the corn row. And what you can see is that we had less biomass in that per unit area in the rye that was planted 15 inches away from the corn row, so the 30-inch drilled rye, and that occurred in both years. I'm not sure if it's easy to see here, but on the right here we have the 30-inch row, 30-inch rye, and then on the left we have the 7.5-inch drilled rye, and this was taken on the 23rd of April 2020. The rye was terminated 3 days later, and then we planted 3 days after that. And so what we noticed was the rye in the 30-inch rows was taller than the rye that was drilled in 7.5-inch rows that was taller than the broadcast rye. So that was kind of an interesting thing to note. So let's look at early corn growth and seedling disease for each of these treatments. So if we start with the corn stand at V1, and what we can see there is that we had the most corn stand where we had the no rye treatment compared to where we had the broadcast treatment in both years. If we look at the growth stage, didn't detect an effective treatment on growth stage in 2019, but in 2020, we did see that the corn that was planted into the broadcast rye or into the 7.5-inch rye was a little bit behind in growth stage. If we look at the disease, radical rot incidence, so this will be the number of plants that had disease. Although there's no significant difference, we can see a trend that in the no rye and the 15-inch rye or 76 centimeter rye, I forgot to change that on my slides. I apologize. So in those 2 treatments, we had less plants showing radical rot than where we had the corn planted into the broadcast rye or the 7.5-inch rye. And then if we look at the severity, didn't see a difference in 2019, but in 2020, we see that we had the most disease where we planted into that broadcast rye or that 7.5-inch rye, and then less disease in the 15-inch rye and similar amount of disease in the no rye. So exactly what we were expecting to see out in the field, mimicking what we saw in that growth chamber. Another interesting thing that we noticed in this trial is just the plant greenness, and so we actually measured this in each year. And there's a photograph there from one of our reps from this year, from 2020, and you can see how the treatments just stand out in terms of greenness. But we went out and we measured the greenness of the plant at VT, and you can see that in both years that there was an effective treatment. So you'll see that the plants were greenest where we had no rye, but then in that 15-inch rye, they were slightly less green than that no rye. But in the broadcast rye and the 7.5-inch rye, those that really had an effect on the greenness of that crop. Okay, so this slide shows the barren ears first, just like we've seen previously. So we have the 2019 data in the light orange, the 2020 in the dark orange. And what you can see there is in the no rye control, we had the least number of barren plants compared to where we planted corn into the broadcast rye. That's where we had the most barren plants. But interestingly, with that 15-inch rye, particularly in 2020, we had a similar number of barren plants compared to that no rye check. And then if we look at the corn yield, so 2019 in the light blue, 2020 in the dark blue, you can see that in both years we had the most yield in that no rye control. But in 2019, we had a similar yield in our 15-inch rye. And in 2020, that yield, although it wasn't similar to what we saw in our no rye control, it was still better than the broadcast and the 7.5-inch rye. So that was very exciting. So that's all I have to share with you today. So let's just go through and summarize some of the things that I talked about. First of all, the corn fungicide trials, or the cardinal trials as I call them, that are done at 6 locations across Iowa. We had negligible disease at all locations and didn't see a yield response from either that V12 application or that R1 application. The trial that I did for using fungicides to control Physoderma was done at the Northern Research Farm with natural inoculum. And what we found was that applications of a fungicide at V6 and V7 and V9 and V10 reduced brown spot and leaf sheath symptoms. We also saw a reduction in the amount of brown spot when we used Xyway in furrow. It's important to note that we didn't see any node rot in this trial, despite using a hybrid that's very susceptible to node rot. So probably had something to do with the drought. And so the question is still out on how fungicides might be used to manage Physoderma node rot. I spent a lot of time talking about 2 cover crop trials that we've been working on. And the goal behind these trials is to reduce the yield drag that can sometimes occur with corn, and just to provide some helpful hints, if you like, for growers who are new to planting cover crops or are considering planting cover crops in 2021. And so one of those trials looked at planting green. Planting green is where you terminate the cover crop after you've planted the corn. And we were concerned about that based on our earlier experiments that showed that we could have more disease the closer the rye was terminated to the corn, and that would reduce our yields. And so what we found in our planting green experiment was we had lower stand counts where we terminated the cover crop after planting corn. Those plants were taller, but they also were lower in weight, so indicating that they were less vigorous. We saw more root rot on those plants, more barren plants, and we also measured lower yields in those planting green treatments. And so, once again, we recommend that especially if you're new to planting cover crops, that you terminate that cover crop 7 to 10 days before you plant that corn. And the more biomass you have with that cover crop, probably the longer the interval you want between terminating the cover crop and planting the corn. And then we also, in the spirit of 2020, although this was started a couple of years ago, we had a social distancing trial. And this is where we planted the corn at various distances from rye. And what we found was that if we drilled the cover crop in 30-inch rows and then planted the corn in between those rows so that there was 15 inches between the corn and the rye residue, that those seedlings had similar vigor to no cover crop. They had less root rot, less barren plants, and they produced similar yields to a no cover crop. And so that was exciting there. One of the questions that obviously needs to be answered is that one of the reasons for planting cover crops is for the water quality, so to improve our water quality, to protect those soils. So how does planting a cover crop in 30-inch rows, how does that affect water quality and reduce soil erosion? The other questions that we have is, can we socially distance and plant green? So there's a lot of work that we still need to do, but for the most part, the further we can get that corn away from the rye, the less disease we're going to see. So I'd like to thank you very much for joining me on this episode for Crops TV. And I will be online so that I can answer any other questions that you have. I would like to say a big thank you to folks in my lab, Giotsna, Sarah, John, Clarice, Rochelle, who actually get out there and do all the hard work. And I also want to say a special thanks to Keith Kohler, Tom Casper, Tom Morman, Andy Lenson, the ISU Research and Demonstration Farm Managers, Stefan Galians with PFI, Sarah Carlson with PFI, Jeff Cernate and Adam Chow with Bayer, and Brent Newberger with FMC for discussions that we had about all of these research that really helped to improve the research. So thanks very much for joining me today, and I look forward to your questions.